Uh, I don't know what to say. The one thing I have to say is that I brought the wrong slides. So it's a lot of science, but as Naomi said, she said, not everybody has to give the same talk. So there's another thing that I want to correct. Everybody thinks that I'm 90 years old, and that rumor is wrong. I am actually 44 years old. I admit to 44. So there you are. So today, I'm going to try to tell you about how does a heart beat. And believe it or not, it's still controversial how the heart, what is the mechanism of pacing. And so I will try to give you some insight into some of these ideas. So here's your heart. This is a human heart, and it has 4 chambers, 2 atria, 2 ventricles. The business really starts at this little blue spot, which is called the SA node, sinoatrial node, and it's about 100 or 200,000 cells, and sends an electrical signal down into the atria through another little station called atrioventricular node, down to the His bundle and Purkinje fibers. Now, each of those cells, if you put a calcium dye in, if there's something that senses calcium, it does exactly that. You see, it's beating spontaneously. And if you look at that cell, which looks like this, really, looks like a neuron, not a heart cell. Here's what a heart cell looks like. This is a huge heart cell, big volume, big area, and these are very small, maybe 1/10 of that size. The other interesting thing in this sort of late 60s was discovered that while these heart cells have an electrical signal that starts from -90 going to +20 and coming down, these cells don't have a resting potential. It's very important to understand how your heart works, that this guy sits at -90 and it's electrically connected to this guy who sits at -55. But they're supposed to be electrically connected. And if they're electrically connected, how is this big guy not bringing this guy down to its level? That's the controversy that still remains. Now, there were, of course, many theories about how this thing worked. And the whole idea was that in the 60s, people discovered that this thing they called a diastolic depolarization. And so the whole game became to understand what was the mechanism of diastolic depolarization. So there were a number of ideas. One of them was turn off of the potassium channels. The other one was activation of a new channel called IF. And the other one was rhythmic release of calcium. Now, from your sort of primary school or chemistry or physics school, you know that inside the cell there's about 100 millimoles of sodium. Outside, there's about 150 millimole, potassium inside, sodium outside. So clearly the forces are for sodium to get in and for potassium to come out. In order to go from -60 to -40, in that area, it means depolarize the cell, there's 2 possibilities. Either you can shut off this channel, that means turn off the potassium channel, so the cell will depolarize, or you can essentially increase the sodium going in, and the thing will depolarize. So therefore, you have these different ideas. And there were two very major physiologists, one working in Oxford, Dennis Noble, and the other one was Wolfgang Trautwein, working in Heidelberg. I worked with this man as a sort of postdoc, and they both decided that, in fact, diastolic depolarization was mediated by turnoff of the potassium. And they had evidence that turned out later on to be wrong, but they thought that this current that was generated, called IF being funny because no current turns on in the negative voltages. Generally, on depolarization, the current goes in. So evidence was that IF reverse direction at equilibrium potential for potassium. That means when potassium was, got beyond the power of the potassium channel, then it actually reversed. So the two of them decided it was turn off of the potassium channel, and I used to teach medical students that it was turn off of the potassium channel. So there were two medical students, young medical students in my lab, and this one became the chief of cardiology as he grew up at UCLA, and he does some in a private practice. And when Jim Weiss was a student in my lab, not in my lab, really at the medical school, he came to me and he said, look, you teach us that diastolic depolarization is mediated by turnoff of the potassium channel, and you have just developed a technique called potassium microelectrode, which means that it can actually chemically sense the potassium. And he said, so can I try taking the pacemaker cells and put the potassium electrode in and show that potassium turns off? I said, I don't work on ASA node, but you can do that. And I said, I don't know how to isolate pacemaker cells. He said, I'll learn how to do that. So he learned how to do that, killed 8 rabbits one afternoon to find where the SA node was, and put the potassium electrode in. And what we found, something that we did not expect at all. So here is, normal, the diastolic depolarization. And here is the potassium electrode. You see, it goes out and then goes back in. And then when we voltage clamp the membrane, what you saw is that potassium continues to come out, or it depletes. There was no turnoff of the potassium channel. So the first assumption was wrong, and then we discovered that the potassium current wasn't turned off, that actually what happened was sodium was going in, and that this current that was carried by sodium was very sensitive to 2 millimole cesium. So 2 millimole cesium blocked it, and it turned out that this was sort of a family of channels known as HCN channel. There's 4 of them, HCN 1, 2, 3, 4, and the one that's in the heart is HCN 4. Okay, so I was young, probably less than 44, and I was invited to give a talk at a big congress like this in Paris. And it was a world IUPS meeting, World Congress of Physiology, and I gave my talk about this pacemaker current being not turn off of the potassium, but a turn on of sodium, and I concluded it was a pacemaker current. This man, who was a professor in Japan, and he was the head of the Physiology Institute at their National Institute of Health, came to me, a very polite man, and he said, you know, I really enjoyed your talk, but I don't believe that IF is the pacemaker. And I don't know, what do you say to a man who tells you, I don't believe you, right? So I said, that's very kind of you. Said, but I would like you to come to Japan for 3 months, and we can do these experiments together. So he had an assistant called Professor Noma, and a medical student from Tokyo, who became later on the chair of pharmacology in Osaka. And I went, and this is in front of that institute in 1982. And this is Jim Weiss, the one that started me in this game, a medical student. This is in Aspen, we're skiing together. So we did this experiment, and we published it in Pfluger's archive. And what we did was something very, very stupid, and anybody can do. So we measured the pacing, and then we had discovered in Philadelphia that 2 millimole cesium blocked the IF. And IF is right in here. You see, we depolarize and hyperpolarize, and we see these currents that were known as the IF current. And if we put cesium, that's blocked. But notice that in presence of cesium, the rate of pacing hardly changes. Hardly changes. And we could not believe it. Anyway, a bunch of experiments, and then we decided, well, if we put adrenaline on a heart, everybody knows that when you run, your heart beats faster, and that's because you have more adrenaline that's leaking into your heart. And so we could easily increase the rate of pacing in presence of cesium, added isopaternal, and the heart speeded up just like ever. But IF didn't decrease. So we concluded that these findings ignored, we concluded that the pacemaker, IF, was not the pacemaker, that it had nothing to do with pacing, that the pacing was related to increasing of the calcium channel current. So we published this paper in a very famous journal, properly reviewed, but they were totally ignored for 24 years, until a Dutchman did the same experiment in 2007. Now, mine were in 1983, and did it in a human SA node, human sinoatrial node, and they discovered exactly the same thing. So now there was a problem. There was a problem because this man here, Dario Di Francesco, a good Italian man who was working with Dennis Noble in Oxford, and that's Susan Noble, the wife of Dennis Noble, that they had cloned this channel. They cloned this channel and turned out to be a totally new channel, and it carried sodium, it carried potassium. And they published this paper saying that this channel carried this current, this IF current. But you notice something if you read original papers, as Sir Andrew Huxley suggested that I do instead of reading reviews. He said, look at this SA node. See, it goes there. This is their records from their paper, right? You see the maximum diastolic potential here is about minus 60, right? And it goes there from minus 60 to minus 40, goes up. But notice that when they try to measure the IF current, it's minus 85, very negative voltages. This current turns on at very, very negative voltages, not at the regular physiological voltages. So what did we conclude? We concluded out of this that in rabbit and human SA node, IF is not the pacemaker current because it's blocked. IF is blocked by cesium and did not suppress pacing of that the cell continued beating. IF is blocked. Then isoproterenol enhanced pacing, right, and increased the pacing rate in IF blocked SA node. That means cesium blocked SA node. So then we concluded diastolic degrowth phase oscillates between minus 55 to minus 40, while IF activates at negative voltages to minus 70. So, and IF kinetics were much too slow. Imagine they were activated at 1.5 seconds, slow, when the heart is beating at 1 to 60 beats per second. So how could a current that's turning on so slowly, does anything to SA node pacing if there's not either voltage is wrong and or its kinetics are slow. So then, puzzling, right? At least the puzzling is that if IF is not the pacemaker current, then why is IF expressed only in the SA and AV nodes and not in the atrium and ventricle? That's the first thing to occur to you, because everybody said, well, if the IF is not the right current for pacing, why is it only in the SA node and IF? Well, why is IF activated only by hyperpolarizing pulses? Very negative voltages. Why is IF encoded by these genes activated slowly, especially in the animals' heartbeats that beat at 600 beats per minute? So, perhaps when everything doesn't work, you say, come up with a new idea. And the new idea is that perhaps IF has a new role. It's not a pacemaker current, but a functional insulator. You can see from these cells, here is an atrial cell, and here is an SA nodal cell. This guy sits at minus 90, big, healthy, athletic, athlete. And here is this little weakling SA nodal cell sitting at minus 50. They are electrically connected, and yet this guy is unable to bring this guy down to its minus 90. So we think that to minimize the voltage mismatch between SA node and the atrial cells, the small SA nodal cell activate a slow depolarizing current called IF when they are hyperpolarized by much larger atrial cells. This allows them to oppose this minus 90 constant atrial hyperpolarizing voltage, in effect functionally insulating them from the negative atrial voltages, but maintaining electrical conductivity between SA node and the rest of the heart. Otherwise, the heart won't work. So then what generates automaticity? So somewhere in 1980s, people start thinking on my favorite topic, which was excitation, contraction, coupling. And some newspaper person in New York asked me, why do you work on excitation, contraction, coupling? I said, because it's a very sexy subject. So in the heart cells, large calcium is released from the sarcoplasmic reticulum for contraction. Huge amount comes out. This rise in cellular calcium activates what we call sodium calcium exchange protein to extrude the cytosolic calcium by exchanging 3 sodium for 1 calcium. That means it generates an inward current. And that inward current is the reason why we have a diastolic depolarization. In the SA nodal cells, this calcium extrusion process generates the inward current is proposed to underlie the diastolic depolarization, and therefore came the concept of calcium clock. Calcium clock. Okay. Now, this man here, Edward Licata, was a wonderful friend, and I think this is at the Gordon Conference meeting, and he actually discovered that if he molecularly tied antibodies to two of the proteins, one of them ryanodine receptor and the other one the sodium-calcium exchanger, he found that they co-expressed nicely in the same area in the SA node. So that was, for him, sufficient evidence that the calcium clock was carried by release of calcium from the SR. So there is the cell you saw beating again by itself. And in somewhere around when I moved to South Carolina, I started working on these cells. These are stem cells derived cardiac myocyte. That means we take a fibroblast, we introduce 4 genes in them, and then we wait and do a few other things, and then the cells start beating, and they're indistinguishable from cardiac myocyte. They have all the same currents, they have all the same calcium release mechanism, but what is good about them is that you can actually, electrophysiologically study them and also introduce new viruses in them carrying other new agents. So here is this iPSC, this stem cell-derived cardiac myocyte beating spontaneously, and it beats, in the culture, beats in the lab, it beats all the time. So what we did in this one, the reason this is a very important slide, is that it is voltage clamped at -50. That means I electrically suppress all the ion currents in it. So it's sitting, beating. First thing it tells you, even for a primary school student, it tells you that those ionic currents didn't have anything to do with it. That this calcium release and uptake occurs as a primordial thing in this SA node. And it is beating. You can do the same thing with the SA nodal cells, and you see the same thing. But this was the first time we did this. And here you can see it, we can quantify it, and you can see in here, I think this thing isn't working anymore. I ran out of battery. Yes. So you see, here is the calcium release, and here is the currents that essentially does that, and it shows in here that in the rat heart, the rates are much higher than in the human, but essentially the game is the same. So we then look at the pharmacology of this beating, right? So notice that unexpectedly, we look at something that suppresses mitochondria and immediately stops them beating. So mitochondria must be involved somehow in release and uptake of this calcium in the oscillation of the pacemaker. Notice in here that we blocked this sarcoplasmic reticulum, the ryanodine receptor. Well, it blocks it also, but not really as effectively as FCCP does. We then tried IF blocker, the one that was cloned by the English-Italian group, and notice that it's insensitive. You add it, and it doesn't care that you have blocked the damn thing. And of course, we block the calcium channel, it immediately stops. So the pharmacology tells us, what is the lessons learned, that these cells are small, spindly shape, with large surface to small volume, making calcium release very effective in these small cells and activates NCX. So that becomes their electrophysiological signature. Molecular signature is the expression of this HCN, HCN2 genes into the IF channel. So, entrepreneurship, as Francis said earlier, Professor Chen says, you know, now something occurs to you. Maybe we could be entrepreneurs and make a biological-based pacemaker. Why one of these battery-driven pacemakers, which the battery runs down, the leads get destroyed, the leads gets infected, and besides, it's an end-of-life type of thing. Why not have a new pacemaker and makes you look like a 44-year-old? And so that's what we did. We took these, we infected these skin fibroblasts from human with these 4 set of genes. We made them into pluripotent stem cells. Then it differentiated the pluripotent stem cells to make them into cardiac myocyte. And then they have wonderful about them is that they have large surfaces, just like the SA node, small volume. They have a robust calcium release and low levels of IK1. That's a potassium channel that keeps the ventricular cells very negative. So just like SA nodal cell, an overexpressed CN4 and SCN2 in them, and then we make a heart cells. And there, voila, there they are. I've cultured them with rat neonatal myocytes, and what you see is they're beating, you see, in a nice way, and contracting and making the heart go. They're driving the heart. So I think that's where I will stop. The people that did this work is my Hong Kong student, Xiaohua, and then Sarah and Tibor, technical student, and my longtime colleague, Lars Schekman from Denmark, and Dr. Wei from Mongolia, that we did this work. Thank you so much.
Thank you very much, Martin. Wow, wow, wow. So actually, this is one of the best examples how discovery fundamental science is important, because the early work you also done in this field provided the knowledge for research in translational way and to solve problems in cardiac pathophysiology and clinics. And policymakers should spend much more money on supporting discovery science. Thank you for all of your efforts. And now the stage is yours, students. Yes. And or anybody, if you have questions, stand up. I think the best way is to ask questions if you stand up, because you will be immediately visible. And before the first students stand up, I could see that you had a pure physiology science in the electric characteristics of the cardiac cells. And now, surprising me, you turn, to a new generation of techniques, technology, biology, everything. For me, I am still in the middle of the whole process. I find extremely difficult for new technologies to learn, and you show just very elegantly, like it would be the easiest way ever can be done for an electrophysiologist. So how did you find these new technologies and how you can apply so effectively?
Yes. Well, that was what I was trying to tell at this morning session to the students, not to be afraid of technology. Technology is your friend. And you're smart enough to really master it, to learn about it. And I'm really, sometimes I think that the rumors about being 90 is correct, that I am 90 because I cannot quite connect things so well together. But even now, I am open to new approaches, to new techniques to solve the problem I have. The big important thing is to have an idea. Now, you think this idea, what I demonstrated here, two things, a very simple idea. Simple idea is what? That the SA node, the sinoatrial node of your heart beating generates a current that's called IF. And nobody really questioned, where does this current activate? What is its kinetics? They just accepted it. It is in the SA node, therefore it has to be, the pacemaker said. I made the same mistake until we could not hide it anymore. When we blocked the current and the preparation continued beating, you couldn't simply just sweep that under the rug, right? So my demonstration, I think, for you at that is to think about the issues that you're working on or the problem. Is it really true what you're being taught or what your professor is telling you? After all, Wolfgang Treitmann was my professor. I learned from him. He was my sort of, mentor. And I used to teach medical students exactly what he told me. But turned out it was wrong. And to this day, medical schools around the country, including probably in Hungary, they teach IF being the pacemaker current. You will not believe it, but it's true. It's coming from a 90-year-old guy, so I'll tell you. So I think the lesson in here is that you need to question it based on what are you telling me this? Just because it's there, it doesn't mean it is doing what you say it's doing. It may be doing something else. And it's amazing, right? When you really think about, you have these 2 billion cells in the heart, huge amount. And these little 100,000 cells, very small cells, are connected to each other. And yet this big iron, big power of the atrial or ventricular cells is not able to connect these small little cells. How could it be? Well, the reason it isn't because when these SA nodes are brought down by the big power of the atrium or ventricle, they have this little secret battery that they turn on, bring it right up. So then they are protected. Because, you couldn't insulate cells against each other, right? Because they're electrically connected. If you insulated the cells, no electricity would go. So a functional insulator it only works in one direction. That was the point.
Thank you. Questions?
Yes. It's a hard concept.
Yes. Any questions from the audience? So what could be then the next step? Now we can see in cellular level how beautifully pacing and moving forward. Can you predict when we can see it in patients?
Yes, I think. So when I went to South Carolina, one of the reasons I went to South Carolina, I was in Washington, the president of the university convinced me that we could really make a biological pacemaker, that they would be, of course, we started doing that, and then, of course, he retired, and I'm still working, and that idea is still there, right? That you, and here is, for example, let's see, which one of these? Ah, yes. But you see in here, we're looking at the SA node and comparing them to iPSCs. And you can see that they act exactly the same thing, same way. So to introduce them, what I think we have proven that these little cells that we have created from stem cells, that they can connect to, to a rat. They're human, they connect to a rat and drive the rat cell. So the next issue is to really do all the clinical stuff, all the clinical trials, that is how robust they are, whether they will stay in the heart that way. So the basic science work has been done. So now the clinicians need to get into the game and essentially make, now, we, Peter, we told NIH that, we could make this thing if he gave us $10 million or $20 million, and they were very excited about the idea. Then they went to the cardiology study section, and the cardiologist said, why is Dr. Morad, he's such a good scientist, why does he want to waste his time on this type of stuff? We already have an SA node. We have already a pacemaker, electronic pacemaker, works fine. So I'll tell you why. So here's something to put in your sort of one of the 2 trillion neurons that exist in your brain. What if you introduce this cell line so that the heart doesn't think as being, beaten to death by some electric pulse coming out of a battery, but it's coming from cells that are totally newly born? Well, the rest of the heart, the trophic factors that we know come out of the heart, come out of the cells, will also rejuvenate the rest of the heart cells, and suddenly you have a heart of a young person, and a young person heart will create a physiological condition which your brain will start working like a young person, brand new brain. So you won't live to be 90 years old, but you'll be 150 years old. And be young, Anyway, that's what I think the excitement is.
Thank you very much, Morad. And we also would like to commemorate your presence in Szeged.